Assalamualaikum, selamat sejahtera, and very good morning for semua. So, okay, I have about 1 hour to talk about nutrition, gene expression, and health disease, which is related to antioxidants, include polyphenols and flavonoids. Okay, my name is Ruzaidi Azli, Mohammed Mokhtar. I'm from Biotech Research Institute. So below is my email and also phone number. So if you have any question after this lecture, you can always contact me through email or WhatsApp. Okay, this is the overview of today's lecture. So first, we will learn about what is nutrigenomics, and then we will learn about gene expressions, especially about the SNPs, single nucleotide polymorphism, and also the epigenetics. And then we learn about what is antioxidant. So in the antioxidant, we have polyphenols, also flavonoids. So, and then we will learn what is the health benefits of this antioxidant, and then how this antioxidant can be used as a personalized nutrition, and last but not least, is conclusion. Okay, now let's talk about what is nutrigenomics. In terms of definition, nutrigenomics is study on the interaction between nutrition and genes. So in this field, normally we study how nutrients influence gene expression and how genetic variation affect nutrient metabolism. So the key concept of nutrigenomics is nutrition can alter gene expression, impacting health and disease outcomes. So what does nutrigenomics tell us? The overall goal of nutrigenomics is to be develop an individual nutrition plan to help achieve and maintain the best health possible. When applied correctly, information related to nutrigenomics and nutrigenetics can even help to prevent, treat, or even cure certain diseases. So we will learn about what kind of diseases uh, normally related to nutrition. In human, there's a variability and variation in terms of genetic factors. So this will lead to differences in nutritional process such as absorption, metabolism, receptor action, and excretion. So this variability and variation affects perception of taste, degree of satiation, and other factors likely to affect food intake. Inborn differences in the activity of enzymes and other functional proteins contribute to variations in nutritional requirement and to the differential interaction of certain nutrients with a genetically determined biochemical and a metabolic factor. So there's a lot of example. For example, uh, some people can taste bitter taste, and some cannot, and some can taste sweetness, and some is cannot. So this is because of uh, genetic variability among human. Next, we move to talk about what is the gene polymorphism. So the term polymorphism means is maniform. So that means this gene can be in many form. So refers to the presence of two or more variants form of a specific DNA sequence that can occur among different individuals or populations. So the most common type of polymorphism involve a variation at single nucleotide, and also called single nucleotide polymorphism or SNP. Other polymorphism can be much larger, involving longer stretch of DNA. But for this lecture, we focus more to SNPs. Okay, for example, here this is a SNPs for two different individuals. So for individual ones, uh, the mother have different DNA sequence compared to father. This is there's a different in base here T and C. So, but for individual two, the mother and father have same DNA sequence. So, for these two individual, of course, there is a different in terms of risk into certain disease. Next, what is the causes of gene polymorphism? So polymorphism are generated by mutation in the genes for this enzyme, which cause decrease, increase, or absent enzyme expression or activity by multiple molecular mechanisms. So for example, here is normally gene polymorphism is caused by mutations. So a mutation is a change in the DNA sequence of an organism. Mutations can result from errors in DNA replication during cell division, exposure to mutagens, and also viral infections. Okay, this is another example of SNP. So this is to show that DNA sequence variation that occur when a single nucleotide, which is ATGC, in the genome sequence is altered. So for example, here is two sequence DNA fragment from two different individuals, which is the other individuals, the single nucleotide is A and the other is a G. So of course, because of the difference is DNA sequence, both individual will produce different product, which is a protein, um, and will affect its metabolism and also eventually will affect their health. So okay, this is the overview of the gene expression. For I'm not sure how familiar you are with the gene expressions. Um, just to recap, but if you already learn about how process of gene expressions, this one is only just to recap how the DNA, RNA is working. So of course, the central dogma that normally we learn about the gene expression is from DNA, it will transcribe to RNA and then translate into protein. So normally this protein will function as a whatever a regulator or maybe to control metabolism and whatsoever. And because of let's say the the DNA is altered in certain people, that's why it will produce different protein or weaker protein. So that's why certain people have a higher risk to get certain diseases. Okay, so because of DNA sequence is quite very important, that's why single nucleotide polymorphism, also called single gene disorder, which is this need can cause certain disease or disorders in humans. So genes involved in disease tendency and susceptibility, and the nutrition environment modifies the expression of genes. Okay, the nutrition cannot um, alter the DNA sequence, but it can alter the gene expressions. I will talk in details later about this. And also the nutrient metabolism may vary by genotype, and this will affect the individual health. So, for example, here I'm not sure whether you can see it or not. So, different between individual one and individual two. So, in individual one, this is the normal individual, which is um, all the process is normal, and then will have the healthy phenotype. But for individual two, there is some mechanism is altered here, and then it cannot go to the normal metabolic activity, so which is the altered metabolic homeostasis, and then that's why this individual is prone to certain disease. Okay, because the metabolism is not normal in this individual too. So a lot of studies already prove that there is a interaction between nutrients and genes. So, for example, here specific nutrients act as regulators. For example, here is vitamins and polyphenols, which is we will learn more details what is polyphenols, and then we will learn about the term epigenetics, which is nutrient can influence DNA methylations and also the histone modification. Okay, because normally nutrient cannot alter the SNPs, but it can change the epigenetics, which is can change the gene expression. Okay, this is an example of a nutrient that can uh, affect the certain genes. Yes, this one is genes for metabolisms. Of course, there's a lot more than this. This is the gene for distribution, and this one is a gene for signal transduction. So, what is the role of nutrition in disease? So, the link between nutrition and disease is when you have a poor nutrition, it can lead to altered gene expression, contributing to disease like obesity, diabetes, and cardiovascular diseases. Okay, nutrigenomic genes, disease prevention. With nutrigenomics, we can tailor the nutrition based on the genetic profiles for personalized diet to prevent or to treat the diseases. Okay, now let's move to antioxidants. Um, we already learned about how nutrients can influence the gene expression, so I will explain a little bit more later. But we will talk about what is antioxidants. So antioxidant is compounds that neutralize free radicals and reduce oxidative stress. This is the keywords here, free radicals and oxidative stress. So this is known quite important in health to protect cells from damage. So normally damage is caused by these free radicals and reduce risk of chronic diseases. So diseases yang biasa uh, related to antioxidant is more to the metabolic disease like diabetes, obesity, cardiovascular disease, cancer, and so on. So types of antioxidants. So for today's lecture, we focus more to polyphenols and flavonoids because this is the most abundant antioxidants, especially in plants. And then we need to learn what is free radicals. So normally when we learn about antioxidants, we need to know also what is a free radical. So free radicals is any atom or atom within a molecules with at least one unpaired electron in its outermost shell or orbital. Okay, this is a free radical. What is the pointer? Okay, so for free radicals, at least there is a one unpaired electron. So why do we want to limit their action? Because of this unpaired electron, it is very reactive, highly reactive. So why this radical is really, really bad to us? Because free radicals can damage membranes, which is lipid, protein, and DNA. So and also it can result in mutation. If it's quite a lot in our body, it can result in a mutation, and also it will alter the DNA sequence, and that's why it's caused different in our single nucleotides. So that's why it's the SNPs terms come out. Okay, when there's a lot of free radicals in our body, of course, it will create the situation, we call it oxidative stress. So this oxidative stress can lead to a lot of diseases in our body. So for example, here is a multi-organ failure, aging, cancer, diabetes, and a lot of diseases can happen because of this oxidative stress. So that's why we did control free radicals and oxidative stress in our body. So how antioxidant can reduce free radicals? So it's very simple. Antioxidant have a lot of electrons. So this antioxidant will donate its electron to neutralize the unpaired electron. So if this unpaired electron is neutralized, this radical basically is neutralized. So normally, it's a free radicals is always produced by our body because of the metabolism, fat metabolism, carbohydrate, metabolism, protein metabolism. So even our respirations, all the activity in our body will produce free radicals. So inside our body also we have mechanism which is the enzymatic antioxidant that can neutralize the free radical that produced by the activity of our body. So normally in the normal state, so we have the redox balance in our body, which is the radicals that produce is can balance by our own antioxidant inside of our body. So, but normally, currently, it's a lot people have this one. It's a lot of more free radicals, but the exogenous, internal antioxidant cannot neutralize the free radicals because it's too much. So that's why normally a lot of people have this condition called oxidative stress. And then if the oxidation is more than the radical, we call it a reductive stress, but this one is quite uncommon. Normally, nowadays, a lot of people have this one, the oxidative stress, which is a lot of radicals because of the activities of our body. And then in our body, we don't produce a lot of antioxidants. That's why nowadays people need the exogenous, means the external antioxidant which is consumed or is get from the our diets and also the nutrients that we eat. Okay, now let's go to what is polyphenols. So polyphenols is of course the key antioxidants. So when we talk about polyphenols, so like it or not, uh, it's always we need to talk about antioxidants. Polyphenol and antioxidants cannot be separated. So a compound containing more than one phenolic hydroxyl group. So I don't want to go more to structure. So the function is to scavenge peroxyl superoxide and also the nitric oxide radicals. So one of the subclass of the polyphenols is we call it flavonoids. So in flavonoids we have flavonols, flavanols, and also anthocyanidin. Contain a phenolic hydroxyl group attached to their ring structure that give antioxidant activity. Okay, normally polyphenol, the source of polyphenol is come from plants, so that's why we call it more to plant polyphenols. So polyphenol is the secondary metabolites. So up until now, there's more than 8,000 polyphenols found uh, worldwide. Yeah? So maybe more than that. More now it's more, maybe more than 10,000. I'm not sure. As I said, the source of polyphenols is normally it's vegetables, fruits, tea, chocolates, beverages, etc. So a lot of studies been conducted and prove that these polyphenols or flavonoids have health benefits. So, for example, it have an anti-inflammatory activity, anti-cancer, cardiovascular protection, and also the neuroprotections. So this is the example of studies that been published. This one is about the polyphenols in mulberry leaf that have anti-diabetic activity, and also this one is the uh, leaf extract, which is have uh, activity in fatty liver, and also this one is the anti-cancer activity of microalgae extract. Of course, in the extract they have polyphenols in the human cancer cell line. So of course, if you Google, if you go to search the journal about the polyphenols and diseases, I think it's more than thousand, maybe hundred thousands of publication already uh, in the database. Okay, this is the other example of health benefit of polyphenols. Again, the list, very long list. I don't want to explain details about this. Of course, basically, if you talk about metabolic disease, people always um, talk about antioxidants and polyphenols. If you see, you read the paper, normally if they study about disease, of course they will study about the content of polyphenols in the sample and also the antioxidant of the sample. So that's because the antioxidant and polyphenols always related to disease. Okay, I don't want to give examples of other people studies. So here I will give examples of my own study actually. So this is the quite recent study, which is I'm studying the mangrove from Sabah. Okay, I we studied I think six species of mangrove in Sabah. So we study in different part, which is leaf, stem, root, and also we extract these mangrove with different solvent, which is from polar to non-polar solvents. So, for example, here we use two type of solvent, which is the ethyl acetate and also the hexane. Ethyl acetate is a semi-polar solvent. Hexane is non-polar solvent. So, as you can see, this is the total phenolic content of the mangrove. So, most of the mangrove samples have quite uh, uh, amount of phenolic content in in the samples. So I can say that all the samples have polyphenols. Okay. Then this shows about the antioxidant of this mangrove. Actually, the lower of value of this IC50 is the higher of antioxidant activity of the sample. So as you can see, not all have a high antioxidant activity, but I think more than half of the sample have quite high antioxidant activity. Okay, that's why normally people will relate with the polyphenols and antioxidant. Normally, if you have antioxidant, the sample also have the high polyphenolic content. Okay, what we do in this study, actually, we evaluate the anti-tuberculosis activity of these mangrove uh, extracts. So, as a result here, I just want to highlight the result. We found that the Syroptaga leaf in ethyl acetate extract have the highest anti-TB activity because this is the IC, which is minimum inhibition concentration. The lower concentration of extract needed, that means the higher of the anti-TB activity. So syrup targa leaf have the lowest MIC value, which is they have the highest TB activity. So then we choose this sample to study about the effect of this extract on the gene expression. Okay, this is the result of treatment of syrup targa leaf uh, in mice. Yeah, so we infected the mice with TB. So this is a gene expression of uh, infected gene, which is TB genes. We choose these two genes to show that the mice is successfully infected with TB. So as you can see here, the healthy one is as a base is 1. So when we infect it with the TB, the gene expression of these two genes increase significantly. So more than 300 times increase. So it's proof that these animals or these mice is infected with TB. And then we do the treatment using in this case is the syrup saga leaf just now. So the rifampicin is the oral drugs, the commercial drugs that have been used in the hospital to treat TB. So this one is as a positive control. We want to compare whether or not it can uh, it can function as good as the commercial drugs that available nowadays. So as a result, the treatment of syrup targa leaf confirm that it can reduce the infection genes uh, of TB. So it's again, of course, for the rifampicin is reduced because, as I said, rifampicin is the commercial drugs that treat the TB. But in this case, our sample also is its proof can reduce also the infections genes related to TB. So then we further, when we already proved the concept that uh, our sample can reduce the infection of TB. So because TB is a if the infection of bacteria, of course, it will involve the immune system. So next, we want to study what is the effect of our sample on the immune genes. So we select here is about 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 immune genes that normally people studied uh, uh, in lung, normally because the samples is we collect it in lungs. So we want to study whether or not our sample can improve the immune genes in uh, TB mice. Yeah. So this one is just to prove that once you infect the mice with TB, so all the genes, the immune genes, will increase significantly. Yeah? Because yeah, there is infection. Of course, these genes will activated and increase. The expression is increased. So most of the genes will increase the expression when you infect the mice with TB. And then this is the result for treatment. So again, with the treatment with the uh, mangrove extract, so it can reduce the the immune gene. That means the immune gene is reduced. That means the infection also been reduced uh, because of the treatment with the mangrove extracts. So it's also comparable with the commercial drugs here. Yeah? So from that we can conclude that the syrup chaga leaf is able to alter the gene expression in TB mice and also improve uh, maybe kill the TB, reduce the infection of TB, and also the improve the the immune gene in TB mice. So okay, next I just want to show the study from my master students, which is she studied in term of the effect of it. Still, we using the mangrove extracts. Here is they use the Bergera gymnorosa roots, and she studied about the effect of these mangrove fractions on diabetes. So this is the result. As you can see here, the aqueous fractions is compared to the diabetic control has significantly reduced. When you treat with the aqueous fraction, the blood glucose is significantly reduced, and then metformin is the commercial drugs to treat diabetes. So as you can see here, the effect is almost similar with the commercial drugs. Okay, okay. And then we also study effect of these fractions on the uh, certain genes that involve in insulin secretion because the sample we take here is from pancreas. So we extract the RNAs from pancreas, and then we study effect of this certain gene. I think we select only three genes here, which is genes that involve in the insulin secretion in pancreas. So the first genes is INS2 genes, which is genes that involve in insulin secretions. Here is the in normal rats, the expressions is of course the baseline is 1. So when we inject with the streptozotocin, which is the chemical that induce diabetes, the expression of INS1 is reduced. And then of course this is because of that's why this uh, mice have diabetes. So what we expect here is when we treat uh, uh, our extract, our sample, this gene expression will increase. So, but what happened here is there is a little bit increase in gene expression of INS2 genes, but it's not really significant. But actually, for the insulin secretion gene, there is another gene which is INS1. So maybe our sample, even here, metformin also is not increased significantly. Maybe the mechanism of how uh, our sample reduce the blood glucose here is not through INS2 genes, but it's more to maybe INS1 genes. But yeah, of course, we don't have that genes in this study. So, but let's look at other genes. Um, um, our sample on the PDX1 genes. PDX1 genes is the genes that involve in the beta cells activity, which is when you increase the PDX gene expression, normally it also eventually will lead to the increase in the insulin secretions. So here again, we need to prove the concept that when you induce the diabetes, the PDX gene will reduce significantly um, when you induce with diabetes in mice. And then, as I said, we expect that uh, when you treat with the samples, these genes will increase significantly compared to the diabetic control. So in our result, as you can see, the our samples able to increase the expression of PDX1 genes significantly compared to diabetic controls. Next is we see the effect of uh, the Bergera gymnosa roots fraction, aqueous fraction on IL-1B genes. So these genes is genes that involved in inflammation in pancreas. So when we induce the diabetes with chemicals, of course, the inflammation will increase in the pancreas. So as you can see here, so the inflammation gene increase in diabetic mice. So when we treat with the our samples, it can reduce the inflammation genes. So this means the inflammation is reduced in pancreas, and of course, uh, as a result, it will the pancreas will working normally and will produce normal insulins. Um, that's why, as a result, the blood glucose in diabetic rats treated with our samples is reduced quite significant. Okay, this is just the function of these genes. Actually, I already told you about this. And then we also run the aqueous fractions to see what is inside of that sample. So using the LCMS. So as a result, we see that most of the compounds is polyphenols, so like catechin, chlorogenic acid, and so on. So that means this maybe this sample is responsible for the anti-diabetic activity in these fractions. So most of these sample actually is already been proved as it's already been studied by other researchers and proved to have the anti-diabetic activities. Yeah. So I already shows the real um, study that prove that these compounds, polyphenols, antioxidant can alter the gene expressions. Okay, so next we will learn how these nutrients or diets alter gene expression. So these actually nutrients can alter gene expression through several mechanisms. Here we propose um, three mechanisms. So the nutrients can, as I said, nutrient cannot alter the DNA sequence. So, but it can alter the gene expression. So we propose here there is three mechanism that how nutrients can alter the gene expression. So first is by acting as ligands for transcription factor receptor. Okay, and then for B, it can metabolized by different metabolic pathway and altering concentrations of metabolites, intermediates, and so on, and will alter the metabolism, and this metabolism will alter the gene expression. And then the third one, they can alter a signal transduction pathway, and then as a result, they will alter the gene expression. So I said previously, nutrients cannot change or alter the SNPs, which is directly because SNPs are fixed genetic variation in DNA sequence itself. However, nutrient can influence how SNPs affect health by interacting with the gene expression and other biological process, which can either mitigate or amplify the effect of certain SNPs. So maybe some of you still confused about what is SNPs or what is epigenetics. As I said, nutrient cannot alter the SNPs. So because normally SNPs are permanent genetic variation. So what nutrients can alter is the only the epigenetics, which is the more to gene expressions. Okay, SNPs affect the the tendency of individual to get certain disease, but nutrients normally cannot affect much in SNPs. So normally, if you talk about nutrients, it will affect more to epigenetics, okay, more to gene expressions and so on. So this is just to give you the table in terms of comparison of SNPs and epigenetics. So hope you can differentiate these two terms there. So we come to I think we come to conclusion of this lecture. What is the practical application of nutrigenomics and also the personalized nutrition? So personalized nutrition is how polyphenols-rich diet can be tailored based on individual genetics profile to enhance health and prevent diseases. So hopefully in future we can create certain maybe polyphenol-rich diets for certain people based on their genetic profiles. Okay. So, genetic testing and nutritional intervention, the future of personalized nutrition and its role in the preventing chronic disease. So, we hope that in future there's a company that can offer this kind of service. Yeah. So, of course, they need to to test the genetic variation or genetic tendency of individuals, and with that information they can tailor a specific diet or specific polyphenol rich diets to that person, so that it can prevent or lower the risk for that people to get disease, to get certain disease. This is hope in future. Okay, to summarize this lecture, um, nutrigenomic studies is the link between diet and gene expressions. So I already talked about what is the effect of diet on gene expressions, and here we talk more about antioxidants. So especially in antioxidants is about the polyphenols and flavonoids play a key role in modulating genes that protect against oxidative stress and inflammations. So the takeaway, a message that you need to remember that nutrition is powerful tool in preventing and managing disease through its influence in on gene expressions. Okay, I think that's all from me. As I said, if you have any questions, you are free to email or WhatsApp me. So of course, you need to introduce yourself first, uh, and then you can ask anything. So hopefully, we can meet in other lectures. I think that lecture is online lecture, so maybe we can have more interactions compared to the video lectures. Okay, thank you very much and good luck.